Kakashi Sensei tells us that one of the scrolls we were gifted included new weapons we can use in training related to the application of concentration cells. So he wants us to focus on 3 different applications. Let's start with the first one, which is measuring pH. So we use a potentiometer to measure pH, similar to experiment 18. So of course we had our glass electrode, which we can now say is a sheet electrode that we continuously put into a solution with an unknown concentration of H+, of course, during our overall titration, right? And now we can kind of throw in layering information and say, well, if we know this is our sheet electrode and we're adding it to this overall solution with a different concentration of H+, we can end up getting 2 half-cell reactions. And so we have our She half-cell reaction here, and then we have our unknown half-cell as well. We can put these 2 together, and we can get our overall reaction here. Now, if we want to get our E of the cell, which is, again, is our non-standard E of the cell, then we can just simply use the Nernst equation. So our non-standard E of the cell is going to be our standard E of the cell, which we know is going to simply be 0 at standard conditions, is equal to negative 0.0592 divided by 2 log of our diluted constant or our unknown concentration divided by our standard, based on our overall reaction here. Now, of course, we'd have to go through and plug in values based on what is given, substitute, and derive. But what we end up getting is that our non-standard E of the cell is equal to 0.0592 volts times the pH. And so our non-standard E of the cell is directly related to pH, which is why we're able to go ahead and use our potentiometer and go ahead and add our electrode directly into our solution, because we're using a concentration cell. Now, how is that possible? Well, the electrode itself also has its own solution relative to the beaker solution that we will be using. And so that's the reason why it's kind of like we have that separation, allows us to get that voltage. Again, we can go ahead and get pH, which we see spit out on a little screen for us. All right, folks, remember that from experiment 18? Okay, now let's get into our second application, which is basically called microanalysis, or using specialized electrodes. So instead of the electrode that we see here for our She electrode, it's kind of like a specialized electrode. And this is like a tip of a needle here. This is one version of it. These are like these tiny, tiny needles that can be used. Now, these specialized electrodes are going to selectively measure ion concentrations in complex mixtures, including very, 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 very tiny amounts. Now, this is going to allow for highly sensitive analysis, like in fields like biology, for example. So this example here is a microelectrode that is recording electrical impulses of a single neuron, which is a nerve cell, right? A brain cell, in a monkey's visual cortex here, which is pretty cool. All right, folks. Now, the electrical potential of a nerve cell is due to the difference in concentration of sodium and potassium ions inside and out of the cell. We see here. So we have our potassium and sodium pumps. You'll learn about that as you learn about neurobiology. Now, some ions are measured with specific ion-specific electrodes. And so, for example, you can have an NH3, NH4 plus electrode that is going to be helping detect industrial wastewater, one for soil and fertilizer, some for blood, some for milk, grain, plant tissues. I mean, of course, for laboratory solutions, whatever it is needed for. And then finally, because neuroscience is such a huge topic, we can specifically focus on the concentration of nerve cells. Again, because we have a difference in concentration of sodium and potassium on the inside and outside of the cells, that again is going to cause an electrical potential, okay? And of course, this electrical potential is going to be used to, of course, send messages throughout the body so we can do things like move, eat, digest, all sorts of things. So nerve cell membranes function as concentration cells, and they utilize enzyme gates, as we can see here, these little gates, which are embedded in the cell membrane, are powered by ATP energy to establish ion gradients, right? And so it's kind of enabling rapid electrical impulse transmission back and forth, which is very important when you're studying neuroscience, of course, and neurobiology. All right, we did it, folks. We can get back to Hokage and get our ranking. Kakashi has told us that we actually got S rank because we had one of the most challenging missions that involved high-level threats, and we actually were able to get through successfully with extraordinary skill. So great job, right? Let's go ahead and rest until we get new missions and lands in the next part. See you there.